The Human Hand in Evolution, an examination of man-made organisms. America has always been a land of innovation, and this spirit extends to how we've shaped the living world around us. From the crops in our fields to the pets in our homes, human intervention has profoundly reshaped life forms, creating what we call man-made organisms. These aren't just natural occurrences. Their very existence and traits are often dependent on us. Our influence on the biological world isn't new. It's evolved from ancient agricultural practices to cutting-edge biotechnological interventions. Think of selective breeding, a cornerstone of human influence since prehistory. It's why wheat, rice, and even our beloved dogs look so different from their wild ancestors today. For millennia, we've been choosing individuals with desirable traits to reproduce, subtly guiding evolution. The term man-made in biology covers a wide spectrum. While selective breeding is a slow, deliberate process, genetic engineering, developed in the 1970s, allows us to directly insert or change DNA in a lab. This marks a significant shift from incidental selection to direct, targeted manipulation, vastly increasing our ability to shape biological life. Methods of human-driven biological alteration. Let's dive into some of the key ways we've created these man-made organisms. One of the oldest methods is selective breeding, also known as artificial selection. It's the intentional development of specific traits in plants and animals by choosing individuals with desirable characteristics to reproduce. Next up is hybridization, which combines different varieties or species to create hybrid offspring. We direct this process to introduce new genetic combinations and traits, often resulting in hybrid vigor, where the offspring are stronger or more robust than either parent. A great example is how maize, or corn, significantly benefited from hybridization with Highland teosinte. This brought hybrid vigor and traits for adapting to diverse environments, helping it become a global staple. Then there's genetic engineering, or GM or GE. This modern biotechnology directly manipulates an organism's genome in a laboratory. It allows us to introduce, remove, or change DNA to express new traits. Unlike conventional breeding, genetic engineering allows for direct, targeted changes and the movement of genetic material across any organism, even creating novel genetic material. Beyond these major techniques, we have a few others that play important roles. Grafting is a horticultural technique where plant tissues are joined to grow as a single plant. Cloning creates an exact genetic replica. And finally, polyploidy involves organisms having more than two sets of chromosomes, which we induce to create specific traits, most notably seedlessness in fruits like watermelons. Man-made plants and foods. Case studies. Our intervention has profoundly reshaped many of the plants and foods we consume daily. Consider maize or corn. It originated from a wild grass called teosinte in southern Mexico around 9,000 years ago. Through selective breeding, we developed larger ears and altered plant structures. A later hybridization about 5,000 years ago introduced hybrid vigor and traits for adapting to diverse environments, turning it into a global staple. Wheat tells a similar story. Wild wheats were harvested in West Asia thousands of years ago, with domestication leading to larger grains and non-shattering heads. Modern hexaploid bread wheat, with its high gluten content, arose from multiple domestication events and subsequent polyploidy, a process involving the multiplication of chromosome sets. Even the banana has a fascinating history. Its earliest domestication occurred around 10,000 years ago in New Guinea, where people exploited naturally occurring seedless individuals. Subsequent hybridization led to the triploid seedless Cavendish cultivars we eat today, which are entirely propagated by human intervention through cloning. And who doesn't love a seedless watermelon? Wild watermelons were originally cultivated for their water content. The sweet, red-fleshed varieties we know today were developed through selective breeding. Seedless watermelons, first developed in Japan in 1939, are triploid hybrids. They're sterile and unable to produce viable seeds, requiring ongoing human technical input to create. Then there's broccoli, a truly remarkable example of artificial selection. It belongs to the same species as cabbage, cauliflower, kale, and Brussels sprouts. All originated from a single wild ancestor, with human selection driving their extreme phenotypic divergence. In the realm of modern genetic engineering, we have Bt corn. This is a genetically modified organism, or GMO, engineered to express proteins from a bacterium called Bacillus thuringiensis, or Bt. These proteins provide continuous, built-in pest protection against certain insects like the European corn borer, reducing the need for external insecticide sprays. Similarly, Roundup-ready soybeans are GMOs engineered to be resistant to the herbicide glyphosate, also known as Roundup. This allows farmers to control weeds without harming the soybean plants, simplifying weed control and potentially increasing yields. Man-made animals. Case studies. Our interventions aren't limited to plants. They extend to the animal kingdom as well. Modern cattle breeds descended from multiple independent domestication events of the wild aurochs around 7,000 to 10,000 years ago. This led to distinct lineages and the global spread of cattle, adapting them to diverse environments through hybridization and interbreeding. Domesticated chickens primarily originated from the red jungle fowl in Southeast Asia around 7,000 to 10,000 years ago. Through selective breeding, we've created birds with larger sizes, increased egg-laying capabilities, reduced fear responses, and a greater willingness to interact with humans. Perhaps one of the most striking examples is the broad-breasted white turkey. Turkeys were domesticated in America as early as 25 AD. Through thousands of generations of selective breeding, farmers prioritized size and meatiness, leading to this specialized bird. This extreme selection has resulted in turkeys unable to fly, prone to obesity-related health problems, and requiring artificial insemination for reproduction. And of course, our furry companions, dogs. Dogs are direct descendants of the gray wolf, with domestication possibly occurring as early as 130,000 years ago. Human-directed breeding, especially in recent centuries, led to the remarkable diversity of modern dog breeds, as well as significant behavioral changes like increased friendliness and adaptation to human society. However, the intense inbreeding for purebreds has concentrated disease-causing genes, leading to breed-specific health problems. And even our ornamental fish have been shaped by human hands. Goldfish were the first fish domesticated for ornamental purposes, originating from the Prussian carp in East Asia around 1,000 years ago. Koi were developed centuries later in Japan from the Amur carp. Centuries of selective breeding transformed them into a variety of colors, body shapes, and fin types for aesthetic appeal. These extreme alterations can lead to health issues, making them dependent on specialized human care. Conclusions. The study of man-made organisms highlights humanity's profound role in shaping the biological world. From ancient selective breeding to modern genetic engineering, we've increasingly manipulated life at fundamental levels. This has yielded significant benefits, like enhanced food security and specialized animals that serve various human needs. However, this intervention also comes with complexities. We see reduced genetic diversity in many of these species, biological compromises in altered organisms, and new ecological pressures from engineered species. Understanding these interactions is crucial for responsible stewardship of our shared biological inheritance, ensuring that our desire to shape nature is balanced with the long-term health and diversity of life on Earth.